Hello everyone and welcome back to the NPTEL course on pharmacognosy and phytochemistry. In the previous sessions, we have seen what are tannins, the different classes of tannins. How do we check whether tannins are present in a given solution? In today's session, we will go through few examples of tannin containing drugs. We will understand the biosynthesis and also applications of tannins. So tannins are biosynthesized in nature from a pathway called as shikimic acid pathway. Now this pathway is unique to plants and microorganisms and does not occur in human beings. The shikimic acid pathway starts with two intermediates, the first one being phosphoenol pyruvic acid, which has its genesis in Embden-Meyerhof pathway, and the second erythrose 4-phosphate, which has its genesis in hexose monophosphate shunt. Now phosphoenol pyruvic acid is a 3-carbon acid, whereas erythrose 4-phosphate is a 4-carbon compound. So this 3 plus 4 carbon compound together kind of cyclize in themselves to give you a 7-carbon compounds, which are called as dihydroshikimic acid and quinic acid. Your tannins have their genesis from this key intermediates, principally quinic acid. So if you see here in your quinic acid, when this hydroxyl group is removed and you have a aromatization of this ring, the structure mimics your gallic acid. Whereas in the previous session, we saw that two gallic acid mutually undergo a lactonization reaction to form what is called as ellagic acid. Now this gallic acid and ellagic acid are hydrolyzable tannins and have their genesis from dihydroshikimic and quinic acid. Now when we go to the condensed tannins, you can see here the shikimic acid pathway also leads to amino acids called as phenylalanine, tyrosine and tryptophan. So let's take a case of phenylalanine. When you see a structure of phenylalanine, you can see here this is essentially a 6-carbon moiety, that is a phenyl moiety attached to 1, 2, 3. So this class of compounds phenylalanine gives rise to are called as phenyl propanoids, that is phenyl plus propane. In addition to that, apart from phenylalanine and tyrosine just differ in a presence of hydroxy group. So when a hydroxy group is added, you get tyrosine. Phenylalanine and tyrosine both give rise to phenyl propanoid moieties. Now how does that go ahead? So if you see the structure of condensed tannins, I can just bring you the same molecule 1, 2, 3, 4, 5, 6. So this is phenyl and 1, 2, 3. So this particular part of flavan 3-ol has their genesis from phenylalanine or tyrosine. Whereas this part, that is the another 6-carbon moiety, has their genesis or biosynthesis from acetyl-CoA as a biosynthetic intermediate. So you can imagine that 3 molecules of acetyl-CoA, this is 1, acetyl-CoA has 2 carbons, so 1, 2, 3. So 3 acetyl-CoA cyclize and then join this phenyl propanoid to give you a flavan 3-ol nucleus. Now this flavan 3-ol nucleus undergo mutual polymerization to form proanthocyanidins, we often refer to as your condensed tannins, the flavanol types. So flavanol type condensed tannins have their genesis coming from your shikimic acid pathway through phenylalanine or tyrosine as a key intermediate. Now distribution of tannins. You can see tannins occur widely in nature. The reason is tannins are defensive molecules. They have protein precipitating As a result, they are very good antimicrobial agents. So they prevent infections in plants. So you will see them principally in bark of plant, where they give you a deep brownish coloration. Those are called as your flobafenes and flobotannins. They occur in cinchona, arjuna, acacia, oak, wild cherry and numerous other barks. If you see the brownish coloration of the bark, that is principally attributed to the presence of tannins. Now when you go to leaves, they may not be brown in color. The reason being the molecular weight here is slightly less. The nature of condensation and nature of tannins present vary. So in leaves you can attribute it to a little astringent taste. So when you drink your tea, that is a decoction, when you have your hamamelis leaf or when you take a taste of your guava leaf, you will get that little astringency which is attributed to the presence of tannins. In fruits, you can have a tart or an astringent taste which is given by amla, pomegranate peels specifically, and berries such as strawberry and raspberry. In seeds, you can see them in coffee. When you drink coffee, you know it's astringent, it has a good pigment value. Cola. Now this cola seeds are also traditionally been used to prepare your Coca-Cola. So those cola nitida or cola seeds. Sal, you have your buckwheat, which is known as kuttu or kutti when you say your buckwheat, or Phagopyrum esculentum. Your supari or betel nut are few examples of seed containing tannins. In fruits, we have amla, which is your Indian gooseberry. We have harda and behda. All three of these fruits put together come in an Ayurvedic formulation. We call it as Triphala Churna. Triphala means three fruits. The three fruits of Triphala are your amla, harda and behda, which are rich in tannins, which are used to treat GI disorders. Certain extracts or concentrates also are rich in tannins. You can take example of your pale and dark katechu. The black katechu is nothing but your katha that you apply on pan, and quinoa, which is your Pterocarpus marsupium plant extract. So let's go to few examples. Now we'll study detailed examples of drugs containing tannins. We're going to go in the details of nutgal, amla, pomegranate peel, myrobalans, especially your harda and behda, and katechu, the pale and black one. If you see the classification of this, the hydrolyzable tannin containing drugs in this are your nutgals, amla, pomegranate, myrobalan. All of this contain tannin derivatives which are chiefly gallotannins or elagitannins, which can be easily hydrolyzed by the enzyme tannase and hence come in the category of hydrolyzable. Whereas if you see condensed tannins, in that category we have few examples such as your pale katechu and black katechu. Now let's start with nutgal as an example. So what is nutgal? So you can see this circular object, that's a nutgal. That's actually a pathological growth, like a tumorous growth, which is found on a very young twig of what is called as Dyer's oak. Now Dyer's oak botanically is Quercus infectoria, or referred to as your oak tree. Now this oak tree is a plant which grows up to a 2 meters height, and what happens here is you will see there are tiny insects. Now these insects lay their eggs. The insects which lay their egg on this oak are your Cynips tinctoria and Adleria galletinctorae. So when these insects lay their eggs, they give rise to tumorous outgrowths. They are called as your galls, and those are then dried and separated. Now geographically, this oak tree is found in Turkey, and Turkish galls are most common ones in market, but they are also found in Syria, Iran, Cyprus and Greece. Apart from that, if you see the nutgalls market, you also come across certain Japanese and Chinese nutgalls. So what are they? We'll discuss in the later part of section, but let's just try to understand what galls is. So when this Dyer's oak or Quercus infectoria, this plant is very susceptible to the insects. So they will lay their eggs, and for the nutrition they tend to lay their eggs on the twig, that is the young part, because the young part is more permeable to the eggs. So as the eggs mature and little larvae come out of that, the larvae starts infecting and feeding on this young twigs. Now when they start feeding on this young twigs, what happens is, in that young twigs you will have starch, and this starch is utilized by this insects by production of something like an amylase. The starch is converted into glucose. Now when the plant senses more and more glucose is being produced in a particular cell by hydrolysis of starch, in that region a pathology changes and abnormal cell division happens, something like a tumorous outgrowth. So this outgrowth continues, and what happens is, as the insect is still consuming these juices which are rich in glucose, the insect tends to get entrapped inside the galls. So you have an insect in center and a tumor like gall growing around it. So slowly initially at the stage of infection, which we call it as stage 1, you will see that this outgrowth is blue or slightly bluish green in coloration, and it starts maturing, it starts enlarging, and as it starts enlarging, you can see here there are certain projections also coming out. So this is the basal stalk. Stalk means the place from which it is attached to the plant. You can see some stalk here. So there's the stalk region, these are all stalk regions, and then you can see numerous circular projections. So here the irregular outgrowth starts of the tumor. All in all, if you see Quercus infectoria galls, they are generally circular in nature. So they will start growing, enlarging till you get a stage 2, which is called as your chrysalis stage. In this, the insect inside is in the chrysalis stage, and you will see the color is still blue or bluish green in coloration. Now stage 3 comes in where your insect still enlarges and goes to a pupal stage. At this pupal stage, it creates and starts digging in the cavity. The reason being, it now wants to escape out from this gall. It no longer wants to remain encaged in it, and it starts boring numerous cavities inside the outgrowth. Now slowly what will happen is, the insect succeeds in boring a cavity, and once it does that, it can escape out of the gall, and once it escapes, you will see some cavities occurring. So during the gradation stage, we can say that initially the galls is blue. Later on, when the insect is mature inside, you will see it growing to slightly olive green, and later on when the insect escapes by making a cavity, it slowly starts becoming grayish or whitish in appearance. Now when you see the pathology of it, initially what happened was, you had lot of starch. This starch was converted into glucose. Then this glucose is being utilized by the insect, but when the plant senses it, it converts and prepares lot of derivatives or secondary metabolites we refer to as tannins. So tannin starts accumulating it in an attempt to kill the insect. So the inner layer of this galls becomes very rich in tannins. What develops or what deposits inside this are your gallic acid. Chiefly this gallic acid goes on polymerizing. So you can see here, I'll just show you with a pen. So you can see here, these are gallic acid esterified with another molecule of gallic acid, and these are all attached to a central glucose moiety. So all this glucose moieties which the insect has produced are now esterified with the help of tannins, especially gallic acid. So you get lot of gallotannic acid, and if you see the chemical composition of galls, approximately 70 to 50% of the chemical composition of gall is chiefly gallotannic acid. Then you have few monomeric gallic acid up to 4 to 2%, ellagic acid, starch. Along with it, inside the fruit, you will have deposits of rosette to prismatic calcium oxalate, and a very characteristic lignin bodies. Because they are very hard, stiff, lignin start depositing in them, and you can easily identify that by staining it with fluoroglucinol water. So if you see the appearance, what is going to happen is you can go, this is like a stalk of it. You will have numerous projections coming out, and if you see the internal cavity, this internal cavity is a large layer of parenchyma. So you have a parenchymatous cells layer out here which are thin walled, and then you might have a single or double layer of sclerenchymatous tissue. Inside the sclerenchymatous tissue is another inner layer of parenchymatous tissue. So I'm just putting here as sclerenchyma and another layer of parenchyma, but the internal parenchymatous layer are thick walled, and they contain lot of this starch, lot of tannins, and this cavity. Now within this cavity you might have your insect if the insect has not escaped, or if the insect has escaped, it appears like a empty cavity in here. And few other compounds also start to be present here. They are little lipophilic ingredients, that is your betulic acid methyl ester and your oleanolic acid methyl ester. So methyl betulate and methyl oleanolate are present along with sitosterol. Now since the plant is undergoing pathogenesis, it will secrete acidic compounds to get rid of the insect, and few of these acidic compounds which the insect secretes are your nictanthic acid, rubric acid, and syringic acid. So a lot of this are present. So if you want to check if the galls are genuine or not, what you can do is just take this galls and dip it in water. The first thing is they should sink. Second thing, if you cut the galls, you should see a cavity inside, and if you add a ferric chloride or any reagent to check for tannins, it should give you a tannin positive test. Now this tannins in galls chiefly are used for pharmaceutical purpose. So in market you will get two type of galls, Turkish galls, and I said earlier you have Chinese or the Japanese galls. The difference between them is the host plant. In your Turkish galls, the host plant is Quercus infectoria, and the insect which generally causes this pathology is your Adleria galtinctore or Synapse tinctoria. Whereas in Chinese galls, it is Rus chinensis, the plant which is generally infected by Aphis, that is Schlectandelia chinensis. Now in terms of chemical differences, here you will find the tannic acid derivative mostly as hepta or hexagalloyl glucose, and in this case you will find mostly as octa or nona galloyl glucose. Now tannic acid is a molecule which has no fixed structure. You can attach as many gallic acid as possible. So in market you get numerous tannic acid derivatives varying in their molecular weight. So by the nature of tannic acid, you can come to know what is the source of this tannic acid. If you see the appearance of Turkish galls, they are more smooth and circular. The photo which I showed you previously were Turkish galls, whereas the Chinese galls are little irregular, knotty, that is they show knot-like appearance or bumps, irregular, and they are soft, that is they break easily, whereas the Turkish ones are little stiff in nature. Talking about the applications, the main applications of these galls happen in pharmaceutical industry because they give you a good quality compound called as tannic acid. Now this tannic acid has a strong astringent properties. This strong astringent properties will help in protein precipitation, and hence it can take care of your wounds. It will close the wounds, stop bleeding, stop oozing, so it can be used as styptic. It can be used to treat minor ulcers. Even in hemorrhoids or bleeding hemorrhoids, they are given as suppositories to kind of seal the skin and stop the bleeding. Now interestingly, one more attribute of tannins is their ability to chelate metals. So initially when there was metal poisoning, a tannic acid solution was administered which could easily, as we saw, chelate the metal ions, and then they could be removed from the system. Also, tannins have a ability to form complexes with nitrogen containing compounds such as proteins and alkaloids. So even in case of alkaloidal poisoning, you will see the nut galls are beneficial as they tend to form complex and precipitates. Now this has been discontinued lately. The reason being, it is found out that despite forming metal complexes, some amount of this tannin still get metabolized in our gut and get absorbed in our system. Once they get absorbed in our system, they were found to give you little adverse effect associated with liver, and that was not preferred. And as a result, what you see now is their decline use in systemic application, but in terms of topical applications, they are used for mouth ulcers, bed sores, topical creams, suppositories, yes. And to the minor extent, they are used in tanning leather. Not the Turkish gulls, but the Chinese gulls also been used in tanning leather and dyeing. Now, along with it, numerous allied drugs are present. We discussed the Chinese and Japanese gulls. Then there are crowned Aleppo gulls. The interesting feature of this is, rather than those projections occurring everywhere, they occur in the apex, that is the top of the fruit, and that is the reason they give you an appearance like a crown, and that's the reason they are called as crown galls. And another ones are Hungarian galls, which are more now cultivated in Yugoslavia, and they are also containing, or they are also rich in tannins and an important source of tannin containing drugs. So we move on to the next drug. The next drug is the Indian gooseberry, or referred to as amla. So this is a common plant occurring in India. It consists of a fruit. It can be used as fresh, or if you even use the dried fruit as mukfas in India, which is coated with or laced with sugar. The plant is Phyllanthus embellica earlier, now it is known as Embellica officinalis, belonging to the Euphorbiaceae family. So this is a plant, you can see by its occurrence, it bears what is called as pinnately compound leaves, and these are little tiny leaflets which occur in pair. You can also see an insect coming out there. Now this is native to deciduous forest of India, Sri Lanka and Myanmar. This plant is commonly cultivated, but majority of this harvest still occurs from wildly grown plants. So the cultivation is done by seeds, it can be done by cuttings, it can be done by budding. This plant give rise to separate male and female flowers. It is seen that the male flowers generally occur here, that is in the leaf axis, whereas the female flowers occur mostly in the stem. So male flowers occur as groups or bunches, whereas the female flowers occur as solitary or single. And once they fertilize, the fertilization rate is about like 30 to 40%. You will see amla mostly occurring here somewhere, because the female flowers are located here on the stem axis. Now the flowering season is generally summers, and post that the fruiting takes a little while, about a 3 months time, so you get that in winter seasons. So chemically, amla is a very rich source of vitamin C. It is said that amla contains almost 160 times vitamin C as compared to your apples. By weight, it contains almost more than 30%, 30 to 40% sugars, about 14% gum and cellulosic, that is a good source of fiber. In terms of a tannin content, it contains chiefly gallic acid. You remember the video we saw in the previous session where we found a blue color complex with ferric chloride? That was the amla solution. So it gives you a nice bluish color complex owing to the presence of high content of gallic acid. It contains ellagic acid and also phylembin as tannins. It also contains alkaloid, that is nitrogen containing compounds such as phylantidin and phylantine. Now amla is commonly taken for diuresis, that is whenever your body swells or there is a water retention in your body, consuming amla helps to get rid of water, especially through urine, then that's the reason it's called as a diuretic. It's cooling for a body. Now having a rich tannin content, it can address hemorrhage, that is bleeding, and it's also used in diarrhea because of its antimicrobial activity. We saw that it's protein precipitating, and it's because of its protein precipitating activity, it binds to microorganisms and kind of effectively remove them. So in Indian traditional medicine, especially in the Ayurvedic medicine, you can see amla being used in Triphala Churna, which is there for GI disorders, and it is also a immune booster because of its vitamin C content, and that's the reason you find it being used in Chawanprash. So moving on to the next drug. The next drug is a very simple pomegranate peel. Now we all have consumed pomegranate, but just in case, when we take a little bite of this peel or that feathery remains of the fruit, you get a little bitter taste, and that's attributed to the presence of polyphenolic compounds, especially tannins. So pomegranate peel is medicinal. It consists of dried peels of Punica granatum, that's your pomegranate, belonging to the family Punicaceae. Now this is a tropical plant which grows in Middle East, a large extent in India and in the Mediterranean region where it is commonly consumed. Now this is a plant which gives you a fruit or flowers throughout. But what the farmers do is they kind of cultivate it in a way that you get seasons to it. So you can curtail water supply to the plant, and in that way, whenever you want the flowering, you could give it abundant fertilizer, give it abundant water, and you get 3 seasons of it. In India, we call it as Ambe Bahar, which is the summer flowering season, generally happens close to January or February. Now when it flowers in this season, the fruiting generally happens in summer, and that's the one which gives you the highest yield. So this is preferred by farmers all over India. The next one is Mrug, or little rainy season, when the flowering season is in June, July, and the fruiting is generally in the September. And the Hasta Bahar is generally post rainy season flowering, and the fruiting happens much later. So of the three, the summer flowering is much preferred due to increased yields. Once in the juice industry, the internal fruit, that is the pulpy seeds, are removed, the peel is a waste product. Now this waste product has now been valorized for its tannin content. So it contains 2 important tannin derivatives. What you can see here is punicallin. Now punicallin is a dimer. Now just carefully see, I am kind of dissecting this, but you can see 2 molecules of hexahydroxy difenic acid. So this 2 molecules of hexahydroxy difenic acid form ester linkage with a sugar moiety to give you punicallin. Now this punicallin further adds 1 more molecule of hexahydroxy difenic acid to give you a punicalagin. So punicallin and punicalagin are the chief tannin derivatives which are found in pomegranate. They further polymerize to give you a large molecular weight compounds also. So in pomegranate peel, you will get your gallotannins, you will get your elagitannins, elagic acid, gallic acid, punicallin and punicalagin. So apart from that, you get flavonoids such as rutin and quercetin, and it is rich in acids such as ferulic acid, syringic, caffeic, coumaric and protocatechuic acid, which are also antioxidants. So this pomegranate peel, if you take it and boil it in a water, it's a very rich elixir of antioxidant compound, and that's the reason it has numerous applications. People have been taking pomegranate peel extracts for its antidiarrheal effect, for its wound healing effect, because it's a tannin rich one, because it's rich in antioxidant. It is known to be good for heart. It is a anti-inflammatory cocktail, you can say. tannins make it a very good antimicrobial preparation, and lot of studies have now been focused towards anti-cancer effect. So pomegranate peel extract nowadays is also proving as a good aid for people with cancers, because this provides you antioxidant compounds and decrease what are called as free radicals in the body. Now this one more interesting finding about pomegranate is, once this pomegranate peel extract is consumed, it is converted by our gut microbiomes in a compound called as urolithins. I just have one structure here, and that's the structure of your urolithin A, but numerous such urolithins have been produced. You can see it is chiefly, if you just put a ring to it, you can imagine your ellagic acid. So it's just kind of a derivative produced by microorganisms coming out of ellagic acid metabolism. Now this urolithin interestingly has good amount of mitocytic activity, like in cases of a malfunctioning mitochondria, it undergoes it pushes it to undergo a phagy, and your mitochondria which are unwanted, which are not working well, are done away with by the body. You can have a anti-aging property because of this. One more thing is, urolithin is also linked to production of energy. So in people, those who want to slim, it is a good source of burning ATP molecules and a good energy burner. So it gives you improved muscle strength. It helps in weight management. The antioxidant property of urolithin is linked to also protective effect on brain, especially people with Parkinsonism have been administered ellagic acids and urolithin. So if you see or go to some common e-websites or e-retail sites, you can see number of urolithin supplements which have come out. But a natural source of urolithin is just you take a concoction or a peel extract of pomegranate that has all the goodness of it. Now pomegranate peel has also ventured into the cosmetic industry because of its very good antioxidant content. It is known for its anti-aging effects. It is known as a sunscreen agent because of the flavin derivatives it carries. It also has other tannin derivatives which decreases the acne. The microorganisms responsible for production of acne are controlled or curtailed with it. So that's also one way to address it. It also addresses the issue of hyperpigmentation, and that's how your pomegranate peel is gaining popularity. You get normal in your markets face sheets, face masks or products containing pomegranate extracts which are loaded with antioxidants. So these are few examples which we will cover. In the next sessions we will be covering your catechu and myrobalance. So thank you everyone, and here are few references for extensive reading of this topic. Thank you once again.